What is ELISA?
ELISA is Enzyme-Linked Immunosorbent Assay.
The first item we find in the kit is the certificate of analysis, where we can check that both the controls and the calibrators have passed the evaluation test. This is done by quality control and is a very important step. The next document we find are the instructions for use, and you can find a lot of very interesting information of the test. So we can find the components, how to prepare the test, what's the diagnostic relevance, general information, how to evaluate the test, and also some scientific reference, among other important information. Microtiter plate. It has 8 rows and 12 columns for a total of 96 wells. This is where the experiment is going to take place. This is the wash buffer. We're going to use this to wash our plate after every different step of the experiment. This is the diluent buffer. We're going to use this at the start of the experiment to dilute our samples in this buffer, and then we can start the experiment. These are the positive and negative controls. The positive one has the red cap, and the negative one has the green cap. These are very important because after we analyze the results, the positive control should be positive and the negative control should be negative. That's how we know that the experiment has gone well. These 5 are the calibrators. They go from 0 to 4, and they are made using different dilutions of a positive serum. So 4 will be the highest and 0 will be the lowest. We use these 5 to form a calibrator's curve with our analysis software, and using that curve, the software can extrapolate a precise result of your sample.
So this is the conjugate that is used for the test. The main primary role of the conjugate is to amplify the signal between antigen and antibody interaction. This is a colorless substrate converted into colored product. Its main role is to produce a detecting signal. This is a stop solution. Its main role is to stop the reaction and then ready for the measurement.